Neural tissue fires aggressively, generating a massive volume of molecular waste. But the brain lacks a traditional lymphatic vascular network to clear this debris away. Look at this anatomical diagram. For decades, the classical model held that cerebrospinal fluid is generated exclusively here in the central ventricular system. The assumption was that it remained strictly on the outside, flowing over the exterior of the brain as a physical cushion. Because the fluid supposedly stayed on the surface, textbooks concluded that deep inside the brain tissue, metabolic waste had to exit exclusively through simple passive diffusion. But the math of passive diffusion does not work for dense brain parenchyma. It is physically too slow to move large neurotoxins through a tightly packed cellular environment. Under a purely diffusion-based model, heavy macromolecules like amyloid-beta and tau protein would inevitably become trapped in the tissue. A passive chemical process cannot prevent the toxic buildup of these specific heavy proteins. The brain requires a macroscopic, mechanically driven clearance system to survive. In 2012, researchers proved the exact that, a hidden, macro-scale fluid network actively operating deep inside the brain parenchyma. They named it the glymphatic system. It is a brain-wide, macroscopic waste clearance network that relies entirely on a type of cell called an astrocyte. The brain compensates for its lack of dedicated lymphatic vessels by co-opting its existing blood vessels, using them as the physical scaffolding to transport fluid. Instead of sitting stagnant on the surface, cerebrospinal fluid is actively driven deep into the tissue along the outside of these arterial walls. Neural waste clearance relies on a mechanically driven plumbing system embedded tightly alongside the brain's internal vasculature. The pathway begins at the surface of the brain. Here, pile arteries rest in the subarachnoid space, completely bathed in cerebrospinal fluid. This cross-section illustrates the pile artery diving through the glia limitans, becoming a penetrating artery inside the dense parenchyma. It drags cerebrospinal fluid down, creating the perivascular space. This tunnel's inner wall is formed by vascular cells, surrounded by a solid wall of astrocyte end feet. Cerebrospinal fluid flows continuously downward from the surface, running inside this isolated periarterial tunnel. By wrapping fluid around the arteries, the perivascular space creates a direct, low-resistance highway, delivering fresh fluid deep into the brain, completely bypassing the strict blood-brain barrier. The fluid is now deep inside the brain, but it remains trapped inside the perivascular space. To clean the tissue, it must cross the outer boundary, the solid wall of astrocyte end feet. This boundary is physically permeable, with precise 20-nanometer clefts between the astrocyte end feet allowing some solutes through, but the primary mechanism is active transport, driven by aquaporin 4, or AQP4, a specific water channel embedded directly into the glial cell membranes. This diagram shows strict functional polarization. The black AQP4 dots are localized almost exclusively on the end feet facing the perivascular space. AQP4 acts as a selective gate, strictly mediating the transport of water and small solutes out of the cerebrospinal fluid into the brain parenchyma. Because the dense AQP4 acts as a biological pressure valve, it drives a massive active influx of fluid directly into the interstitium. Astrocytes effectively form a regulated biological gate. They allow pressurized fluid to flush deep into the tissue while keeping the brain structure intact. Once inside, the fluid undergoes convective bulk flow. As shown in this schematic, the fresh cerebrospinal fluid physically mixes with the stale interstitial fluid, filling the beige space between the neurons. This deep tissue convection requires a mechanical pump. That pump is arterial pulsation, driven directly by the cardiovascular heartbeat, sending pressure waves into the tissue. This pressurized, mixed fluid sweeps through the parenchyma, picking up heavy metabolic waste, specifically neurotoxic proteins like amyloid-beta and alpha-synuclein. The waste-heavy fluid is pushed completely out through perivenous clearance pathways, flowing outward along the brain's veins. Once it reaches the surface, the fluid drains out of the skull entirely, channeling into the meningeal lymphatic vessels and the nerve sheaths surrounding the head and face. The waste ultimately deposits into the deep cervical lymph nodes, where it joins the rest of the body for systemic breakdown and removal. Driven by arterial pulsation, the interstitial spaces act as an active pressure washing system, clearing neurotoxins. Glymphatic transport does not operate at a constant biological baseline.
The volume of fluid moving through the brain is heavily state-dependent. During a wakeful state, the brain secretes neuromodulators like norepinephrine. This chemical signal causes both neuronal and non-neuronal cell bodies to physically expand. Expanded cells tighten the interstitial space, choking off the influx of fresh cerebrospinal fluid. Sleep reverses this. Lower norepinephrine shrinks the cellular matrix, widening pathways so fluid rushes through. During sleep, the brain's interstitial space expands from 15 up to 24%. That 60% volumetric expansion directly correlates to an exponential increase in convective fluid flow and the rapid clearance of metabolic proteins. Sleep provides the exact physical dimensions required to run the brain's plumbing. It is a strict biomechanical imperative. When this precise fluid dynamic architecture breaks down, the physical consequences for the brain are severe. Triggers like natural aging, traumatic brain injury, and ischemic stroke severely disrupt the clearance system. These events cause AQP4 depolarization. Normally, AQP4 channels align exclusively at the perivascular endfoot. During depolarization, they detach and scatter randomly across the astrocyte's body. Without this strictly polarized directional valve, the pressure differential vanishes. Glymphatic bulk flow stalls almost completely. In this stagnant fluid, toxic monomers like amyloid-beta are no longer flushed away. They drift, collide, and begin irreversibly clumping together into dense, insoluble plaques. This malfunction in macroscopic fluid transport reliably precedes the significant amyloid-beta deposits used to diagnose Alzheimer's disease. Defeating neurodegenerative diseases requires treating the human brain as an interconnected, fluid dynamic ecosystem where mechanical flow dictates chemical survival.